Hi, today I will be talking about the Down syndrome. Down syndrome is a common genetic disorder that causes intellectual disability and characteristic physical features. It's normally caused by extra genetic materials or misallocation of genetic materials. Now, let's delve into it. So about Down syndrome, so this is a normal cell, right? So we store our genetic information in the nucleus, and later on these genes turns to be chromosomes. So in a normal cell, we have 23 pairs of chromosomes. So the most common causes for Down syndrome is the first one is called extra 21. So people without Down syndrome normally have two strands in the 21st pair of chromosomes, but people with Down syndrome has an extra strand in this pair, and this extra copy is called trisomy 21, and that causes some problems. So the other common type of Down syndrome is called translocation. This one is inheritable. Actually, the 21st pair chromosome is totally fine for this one, but the 14th pair normally has some extra material down here that causes some problems. So now we know the causes for Down syndromes. Let's move to the common symptoms. The first thing for Down syndromes is that people with Down syndrome has intellectual disability. So their IQ stays at the level of an 8 to 9-year-old. And the second feature is called craniofacial feature. So what's going on is people with craniofacial features has epicanthic eye fold and has flatter nose. The third part of Down syndromes, which is a common feature, is that the other part of the body, for example, the hand, for people with Down syndrome, this line here is continuous and it doesn't break. But for normal people, this line is actually just split apart around here. So that's the other thing. The last thing is that people with Down syndrome has problems with their heart. So in our normal heart, we have some heart walls here that split this part up, and so blood doesn't get mixed. For people with Down syndrome, this part actually does not exist. So the blood mix-ups that cause many serious problems. So I hope this video has explained Down syndromes. This is a very brief overview of everything, and I hope this helps.
Alright, so now I'll be talking about Huntington's disease. It was first described by George Huntington in 1872 in Long Island, and finally in 1993 we were able to discover the gene that causes it. So by definition, it is a fatal genetic disorder that causes the progressive breakdown of nerve cells in the brain. Some of the effects, it just deteriorates a person's physical and mental abilities. About 9% of cases tend to sadly end in suicide, but most die between 15 and 20 years after diagnosis. Currently, there is no cure. As it says above, it is a fatal genetic disorder, but hopefully soon enough, through technology, we'll be able to find one. So its symptoms at first can be described as like having ALS, Parkinson's, and Alzheimer's disease like simultaneously, but like more specifically, personality changes, mood swings, depression, forgetfulness, impaired judgment, unsteady gait, and involuntary movements, slurred speech, difficulty in swallowing, significant weight loss. And it tends to appear between ages 30 and 50. So now we can look at some of the causes. First of all, if a parent has it first, their child can inherit it, and it tends to be at a 50-50 average. So it's a very high possibility that if a parent has it, their kid would have it as well. And scientifically, it's caused by a hereditary neurodegenerative disorder, which is caused by an expansion of repeating C, A, and G codons triplet series in the Huntington gene in chromosome number 4, which we'll see a picture of after. The full function of it is not known, but once it is paired with the repeating triplet, it seems to be like somewhat toxic to brain cells. And then here in this picture, we can see kind of what it looks like. You have part of the chromosome as it shows here, and then if we zoom in over here, you'll see the DNA strand and you'll see the repeating series.
Hey guys, now we're going to talk about cystic fibrosis. It's a hereditary disease that affects the lungs and digestive system. So let's take a look at why and how exactly it does this.
Someone who doesn't have cystic fibrosis has a lung cell surface that
looks like this. They contain a CFTR gene, which is responsible for transferring chloride ions in and out of the cell. As you can see here, these dots resemble chloride ions. So for someone who doesn't have cystic fibrosis, these chloride ions are able to go in and out of the cell with ease due to the CFTR gene. This means that there will be a balance of chloride ions inside and outside of the cell, which maintains the right amount of water and salt ions in the mucus lining of the lung airway. The additional mucus is swept away by the cilia, keeping the surface of the lung cell clean. However, with people who have cystic fibrosis, this is not the case. As mentioned, the CFTR gene is responsible for transferring chloride ions in and out of the cell. However, with someone with cystic fibrosis, they contain a defective mutant CFTR gene, which weakens the ability to transfer chloride ions in and out of the cell. As a result, chloride ions concentrate inside cells and draw in water by osmosis. With less water outside the cell, the mucus becomes thicker, blocking the cilia, which means that the additional mucus is not able to be swept away by the cilia, which results in a buildup of mucus. This has severe ramifications. Additional blockage of mucus can lead to a variety of symptoms such as shortness of breath, persistent coughing, and an increased risk of lung infections such as bronchitis and pneumonia. There's no current cure, however, treatments such as airway clearance techniques can free up mucus. However, with the increased importance of genetics and bioengineering, researchers are looking at how gene editing can play a role in finding a cure to cystic fibrosis. Who knows, maybe in the next 5 years, we may have a cure.
Inherited genetic disease is sickle cell anemia. It is the most common inherited blood disorder in the United States, affecting 70,000 to 80,000 Americans. How does it work? This disease affects the hemoglobin, which is a molecule in red blood cells that delivers oxygen to cells throughout the body. The hemoglobin consists of 4 protein subunits. 2 of these subunits are called beta globin. When someone has sickle cell anemia, there has been a mutation in their HBB gene, leading to a different version of the beta globin subunits. The HBB gene provides instructions for making beta globin and is found in chromosome 11. This mutation leads to hemoglobin S. In people with sickle cell anemia, hemoglobin S replaces both beta globin subunits. These abnormal beta globins lead to blood cells that are sickle-shaped. Sickle-shaped red blood cells die earlier than normal blood cells, which can lead to anemia. They can also block blood vessels. These deformed red blood cells can lead to a lack of oxygen flow to parts of the body, leading to fatigue, pain, and swelling of the hands and feet. This disease is inherited in an autosomal recessive pattern. This means that the individual with sickle cell anemia has both copies of the mutated gene in each cell. The parents of this individual must each carry 1 copy of the mutated gene. When both parents have the genetic defect, there is a 25% chance that each child will be born with sickle cell disease. Before, people with sickle cell anemia were not expected to live past childhood, and now half live past 50. Some treatments include antibiotics, pain management drugs, and blood transfusions. There is also a new drug called hydroxyurea, an anti-tumor drug which helps prevent the sickling of blood cells. The only current cure is bone marrow transplant. But in order to have success with this treatment, the individual needs a compatible sibling donor, and only 18% of children with sickle cell disease have a healthy sibling match. This treatment can also be risky and have complications. Gene therapy could also be a future cure. Currently, scientists are experimenting with correcting the defective gene and inserting it into the bone marrow of the sick person to stimulate production of normal hemoglobin.
Today I'm going to talk to you about hemophilia and the genetics behind hemophilia. So hemophilia is going to be a disorder that slows the blood clotting process. So that means that anytime someone with hemophilia gets a scrape or an injury, or they have surgery, or they even have a tooth pulled or anything like that, that means they're going to experience prolonged bleeding or oozing just because they have a problem with the way their blood clots. And so there are going to be 2 major types of hemophilia. There's going to be hemophilia A, and that's going to be known as your classic hemophilia, and then there's going to be hemophilia B. And so the symptoms of these 2 and the way they manifest is going to be very similar, but there is a difference in the genes and the gene mutations related to the 2 types of hemophilia. So we're going to take a look at those now. So changes in the F8 gene are going to be responsible for hemophilia A, and the F8 gene is going to be that gene right there that you see the arrow pointing to. And so the F8 gene provides instructions for making a protein called the coagulation factor 8. And so then we're going to look at the F9 gene, which is this one right here, just a little bit over. And the F9 gene is going to be the one that's responsible for hemophilia B. And so that one is responsible for the coagulation factor 9. So it's just the factor 8, and then the factor 9 is going to be the difference between the two. And then, so hemophilia is going to be an X-linked recessive gene. So that means that it's very rare for females to have, just because it would have to affect both of their X genes. And so as you can see here, if you have an affected father, you're likely to have only infected daughters. Or if you have an infected mother, then you could have either an affected son or an affected daughter. Thank you for watching.